Well, I think it's time to start our second session, and this will be devoted to EMBL. And our first speaker is Wojciech Galej, and he is a group leader in EMBL Grenoble, and he will talk about science. So the title of his talk is Structural Studies on the Splicing Machinery.
Okay, thanks very much for having me here. It's a great pleasure to be at this meeting. So I'm a group leader at EMBL Grenoble, and over the next half an hour I will try to tell you a little bit about my experience, and I will try to answer a couple of questions. Does it work? Can I flip slides? Yes, it works. Okay. So I want to answer a couple of questions. So first, I want to tell you who I am and why actually I'm here, and what are my credentials to talk to you about this. Then I will tell you a bit about my scientific story and how I ended up at the EMBL. And then I will introduce the EMBL and tell you in more details what really EMBL is. And finally, I want to just mention what does it all mean to you, and what are the benefits for Polish researchers because of our membership at the EMBL. Okay, so I did my undergraduate degree at the University of Warsaw, and I graduated in chemistry and molecular biology, after which I moved to Cambridge, to MSc Laboratory of Molecular Biology, where I did my PhD studies. And after that, I stayed for a short postdoctoral research. This is something that on any career workshop, people would advise against, to stay in the same lab for the postdoctoral research, but there was a very good reason for that, and there was the time when the newly emerging electron microscopy technologies were coming to life, and I really wanted to learn that. That's why I stayed there. And after that, I was recruited to EMBL in Grenoble, where I'm ever since. Okay, so one thing which was mentioned already before is that, people said that it is very important to go abroad for short internships, and I really have to say that during my undergraduate studies, I went to Cambridge for a short two months summer internship, which really kind of influenced my entire career, because after that I returned to the same lab, and actually it really set my career on a track for the next 10 years. So that was really important for my scientific career. Okay, so what I was doing there, I was studying a process which is known as the pre-mRNA splicing. So for those of you who know about biology, eukaryotic genes contain introns. Introns are those non-coding sequences in between coding sequences called exons, and they have to be removed from precursors of messenger RNAs, and that happens in the two-step transesterification reaction. So in the first step, we have this very characteristic intermediate, and then the second step, spliced exons are produced, which are then engaged with the ribosome to produce proteins. So really, this is the message which is expressed during the gene expression. Okay, so this is process which seems very simple. So there's from the chemical point of view, these are just two simple reactions, but there's enormously complex molecular machinery which is involved in coordinating it in time and space. And this machinery is known as the spliceosome. So I'm not going to go through nitty-gritty details of the splicing cycle, because I'm sure that you are not particularly interested in that. I just wanted to emphasize that this is a very complicated process. My slides are a bit changed, so these are all different subunits that are very faint here. But splicing process is complicated. The whole machinery assembles de novo for each individual intron from many different subunits. There's more than 100 proteins which are involved in this process. And all those individual building blocks of the splicing machinery are also very, very complex. And this is known as the splicing cycle. There are 4 different phases of the splicing cycle. And right, because of this very dynamic nature and on very many different intermediates, the structural studies of this system were very difficult until very recently. So I wanted to show you a couple of examples of research that has been done on this system before I was joining the group. So this is the first electron microscopy images of the spliceosomes purified from human cells. And the reason I'm showing this is it's taken from a paper that was published in the year 2002. In the year 2002, we already had high-resolution structures of the ribosomes, RNA polymerases, all sort of machineries which are involved in a central dogma of expression of genetic information were quite well understood. However, spliceosome was lagging behind by about 20 or 30 years. So we really didn't know very much how does it look like, this machinery which catalyzes that reaction. Over the years, people made some progress. So we have here a couple of examples of the electron microscopy reconstructions of these individual intermediates in the assembly process. But what we really can see here is only a shape of the molecule. So the people use electron microscopy, but they could only see very low resolution shape of the molecule, how it looks like. And for everyone who thinks about biology in terms of chemistry and would like to understand the mechanism, this really is not enough to understand what is happening at the heart of the machinery. So for that very reason, people spent much more effort during those days studying spliceosomes fragments using X-ray crystallography. So at that time, X-ray crystallography was the leading method in structural biology because it could produce high-resolution structures. And the only problem with X-ray crystallography is that you need to have crystals. So you need to take your biological molecules and pack them very neatly in three-dimensional arrays that would have some periodicity. And then we use very, very brilliant X-ray radiation to make a diffraction experiments of those crystals. Now, if the molecule is very big and flexible, it doesn't really want to form a crystal, and that's the major bottleneck for X-ray crystallography. That's why for many years it was restricted only to small fragments and not the big machines. So this is an example of the work which I did on using X-ray crystallography on the spliceosomes fragment. So that's work which comes from my PhD project. And we solved the structure of actually two proteins from the spliceosome to very high resolution, and you can see how it looks here. And this protein was very important because it is kind of positioned at the very heart of the machinery, and I will come back to that. Okay, so here's the problem with the crystallography. So that was a very challenging project, and I was lucky enough to be there at the right time in the right place. Even though this is only a binary complex with 220 kilodalton, I think the whole effort of the entire lab that went into this project was probably about 10 years. So really this kind of research, and I want to emphasize it, is only possible in the places which promote long-term funding. And usually it would be very difficult to do this long kind of project based on the grants. Anyway, we wanted to move forward, and we wanted to study the entire machinery, so those things which I showed you in the cycle. And the problem comes with the fact that it was much more complex. So there we had about 50 proteins and more than 10 times more mass of proteins. So it was very clear at that time that we needed some different approach, that it won't be possible to crystallize the entire spliceosome. And one would say that we needed some kind of revolutionary approach to that. And indeed, there was some small revolution in the structural biology which happened more or less at that time. So what happened, basically people developed new kinds of detectors for electron microscopy. So these are detectors which are much more sensitive, they are much faster, and they really produce super high quality images. And then this in combination with the better microscopes and also statistical image processing, all those developments allowed to visualize very flexible and dynamic macromolecules at the level of details which was unprecedented before. So this really was a big improvement, and it was actually awarded a Nobel Prize in Chemistry in 2017. And what I think is worth mentioning that Jacques Duboucher, one of the recipients of the Nobel Prize, did all his work for this Nobel Prize being at the EMBL at the time. And another person was working at MRC-LMB and Rockefeller University. Okay, so what is electron microscopy for those of you who don't know? So it is a method which from the technical point of view, it relies on putting our sample on a small, about 3 millimeter in diameter grid, which is then blotted with filter paper and then plunged into liquid ethane, which has very low temperature. And this is done in order to preserve specimen, to freeze it. However, the freezing is so fast that it does not form ice crystals, and that's the whole point of doing it this way. Now, once we have a frozen specimen, this grid, Usually we're aiming to get molecules suspended in ice in the holes on the grid. So these are individual particles which we want to image. They are frozen, but inside the amorphous ice. And what we do, we insert the sample in the microscope, and then electron beam comes through the specimen, and it is transmission type microscopy. And we record images at the detector at the bottom of the microscope. And this really allows to produce a lot of data, which are essentially two-dimensional projections of three-dimensional objects. And this data can be used together with some computational methods to reconstruct three-dimensional pictures of the objects of interest. So as a structural biologist, this is our goal, to obtain pictures and shapes and insights into details of molecular machines. Thanks to this method, the picture which I showed you before, this complicated splicing cycle, which was very poorly characterized before, was completely transformed within 4 years. So as we speak now, we have a very good idea about the structures and the mechanism of most of these assembly intermediates and transitions between different steps of splicing, which really revolutionized the field of splicing as well. So, yeah, and this wouldn't be possible without people who actually were driving the progress in the splicing field. Those are 3 people who are running big labs doing research on the cryo-electron microscopy of the splicing machinery, and I was part of Kiyoshi Nagai's lab at MRC LNB in Cambridge. Okay, so I will just try to show you a couple of insights that we got from those structures, from the spliceosomes. So here's how it looks at the very early stages. So there are a couple of complexes which contains many different proteins and RNA molecules, and those are really defining intron-exon boundaries. And those 2 molecules come together at some point. But first I will focus on one of these complexes. Without going into many details, I just wanted to show you what kind of information we get from electron microscopy experiment. So we obtain electron density maps, which actually can be interpreted in a lot of details, and you can see here secondary structure elements for proteins, but also for RNAs. And this particle contains 30 different proteins and 3 RNAs, and this is the biggest building block of the splicing machinery. Okay, so these 2 particles, they come together to form the entire spliceosome. And they're really binding one on top of another. And yeah, I'm sorry, you can't really see very, very well the outline of the whole complex. But this is the spliceosome, which is formed, which is inactive. So that's the molecule which contains all different subunits that are necessary for the reactions which I showed before. However, it cannot do any chemistry because the active site is not formed. So we solved a couple of structures which actually visualize what happens during the transition from inactive to active spliceosomes. And this is just to give you an idea without going into details. What happens? Well, the network of RNA-RNA interactions inside the complex is completely remodeled, and that happens by the action of a couple of RNA helicases, which are disrupting some of the interactions, and they are reforming new ones. And when it happens, the overall structure of the entire complex changes. So you can see here there's a complex which was already remodeled. During this process, about 20 proteins are ejected, and about 20 other proteins are recruited to the spliceosomes, and some RNAs are also lost. And thanks to those rearrangements, we can form the active site in the splicing machinery. Okay, and later on, there are a couple of other events which are happening. For example, a substrate which is embedded in this machine is there. However, the reactive components of the substrates are very far away from each other. And in order to do splicing reaction, we have to bring both parts which are going to react together to the close proximity. And that is catalyzed by some other helicases, and that leads to the formation of another intermediate in the cycle. Okay, so this is a zoom in into the active site. So this, again, just to visualize what kind of information we get from those cryo-EM studies. And here we have a network of the RNA molecules at the very heart of the spliceosome. So there are 5 different RNAs which are coming together and forming this active site. And the intron and exons are visualized here. And what was interesting, we solved, as I mentioned, we and others determined several structures of many different intermediates, and we started getting a picture how the whole machinery works. For example, you can see here that there is a big rearrangement between two different steps of splicing, which is there because the spliceosome is a big machine. It catalyzes two different reactions. However, it has only one active site. So the active site has to be vacated between first and the second step. And it really happens here. You can see beautifully when you compare those two different structures that the branch helix, so-called by splicing scientists, is undocked from the active site, forming a space where we can introduce the second step reactant, which is 3' exon. So it's really quite remarkable how these electron microscopy studies helped us to visualize the whole process at the atomic level of details. Okay, so I will finish that part of the talk now, and that was something which I was doing when I was still a postdoc. And I moved from Cambridge to Grenoble to become independent investigator at the EMBL. So with this, I kind of told you how did I end up at the EMBL, and now I'm going to tell you what really is EMBL. So EMBL is was already mentioned, it is an intergovernmental organization. It's an international institute which was formed from the initiative of John Kendry and Jim Watson. And it was really meant to promote life sciences in Europe. And it was meant to become a center of excellence that would do research which would be comparable to the quality of research that was being done in the US or in the UK at that time. Because this kind of stuff was missing in Europe. Europe was not, in the mid-70s, Europe was not as competitive as some other places, at least in terms of molecular biology. And as I mentioned, EMBL was founded as an intergovernmental multilateral treaty, which really means that it is not meant to benefit any of the individual countries, but it is rather meant to benefit all member states as a whole. And these days, it has grown much bigger, and instead of 10, we have 26 member states. Poland became a full member state only last year, and I think the latest member state is Lithuania, which joined only, I think, a couple of days ago. Yeah, so even though it grew very much in the size, so we have these 26 countries, some of them are not even in Europe. Yeah, the lab itself physically is located in six different places. So the main EMBL headquarters is located in Heidelberg, and there are many different kinds of life sciences which are happening there. There are two sites which are dedicated to structural biology, which is EMBL in Grenoble, where I'm based, and also in Hamburg. And traditionally, those sites were located close to the synchrotrons, which are producing X-ray radiation, and that is used for X-ray crystallography of proteins. So it's kind of experiment I showed you before. That's why they were there. There is EMBL EBI Bioinformatics Institute close to Cambridge in Hingston. And there are also two other outstations, now there are units. So one in Rome, which deals with epigenetics and neurobiology. And another one in Barcelona, which specializes in tissue biology, and it was opened only very recently, a couple of years ago. Okay, so even though EMBL was founded already in the 70s, and it has grown significantly since then, the basic principles on which it operates remains the same. So it really means that it puts a lot of emphasis on scientific excellence, diversity, scientific freedom. It really promotes collaboration between different people with different expertise. It supports cutting-edge infrastructure, as well as some of the rules are like staff turnover, which was mentioned earlier, which really means that everyone can stay at the EMBL only for a certain period of time, which is 9 years. And regardless of the scientific output, most of the people are forced to leave after that time. And the EMBL also promotes independence at the early stages of the career. Okay, so these are the main missions of the EMBL as a research lab. So EMBL promotes basic research by allowing people to do really kind of forefront science in biomedical science. It also provides a lot of services, and I think this is very, very big part of EMBL is to to provide services not only to people who work at the laboratory, but also make them available to the outside, so everyone can use them. Another thing which is now a status is training. So EMBL is meant to train and inspire next generation of scientists, which is a very important mission. Technology transfer is always a big deal at the EMBL, and there's a lot of cutting-edge technologies which are being developed all the time. And finally, it is meant to integrate life sciences. Okay, so going a little bit more into details, there are about 80 independent research groups at the EMBL, which are located in 6 different sites in 10 different units. So these are all units which are at EMBL in Heidelberg. Some of them are doing some advanced light microscopy experiments. For example, this biophysics is very much focused on that. There's developmental biology unit in Heidelberg as well, genome biology, as well as structural computational biology unit, which does a lot of people there that do kind of research which I just presented to you. And the director research unit is a place where director general, deputy director, and EMBO director are having their own groups doing research. Now, I mentioned structural biology units at Hamburg and Grenoble here. And again, there's bioinformatics and epigenetics, as well as tissue biology. Okay, so in addition to doing all these mission statements and doing science, EMBL provides a lot of services which are available to external users. And one of those services is, for example, crystallization services, which are run, among others, at the EMBL in Grenoble. Bioinformatics services, which I think if any of you have done any kind of molecular biology before, I mean, you must have used EMBL EBI platforms, which are really widely used, about 27 million hits every day, which is quite impressive. And core facilities are integral part of EMBL, which are very important there. And there are many of different core facilities, and this is really great thing about the EMBL, because they provide kind of state-of-the-art instrumentation which is available to everyone who wants to use it, together with the staff members who are actually helping to analyze data, and they really act as a scientist, almost like a collaborator, to help to analyze data and squeeze most out of it. And this is not everywhere a common practice, but this really makes a huge difference, and then myself and my group is using those different core facilities quite a lot. There are 2 projects at the EMBL which are very big at the moment. So one is a mesoscopic imaging facility in Barcelona, but something which we are all very excited about is bioimaging center, which is planned to be placed in Heidelberg, and actually the construction already started. So this is going to be an imaging center which will host all different kinds of light and electron microscopy. And what is very important about this is it will be at the scale which is kind of unprecedented in Europe. There will be really huge facility which will be available to European community and member states. So this is a flagship project of EMBL at the moment, and we are really looking forward to have it finalized. Okay, so a few words of why should you really care about all these things, because this is one of the reasons why we are here. So thanks to the full membership of Poland in the EMBL, which happened only last year, we have better access to core facilities and the services. So all these core facilities and services which I mentioned earlier, people from Poland as a member state can have access to that, and then they should really make use of it. The other thing is that we can participate, people in Poland can participate more proactively in different kind of collaborative efforts with EMBL. They can form partnerships, and there's a great example of this Brain City, which is a partnership between Gdański Institute and the EMBL, which I will come back to that. And of course, there's access to different platforms, and the EMBL is not only about the research labs, it's also about the community, and being a member state, we have access to the network of EMBL, which is very, very important. And then finally, things like technology transfer. We have also a lot of expertise, and people are happy to share this stuff. So this is one slide which I was given also to show, which is about this brain city. So that's a partnership between EMBL and Gdański Institute, and that's really exciting new collaboration, which is based on this MAP project. And I think Leszek was here, is leading this from Polish side. And yeah, we are all excited, and we are looking forward to see what will come out of that. Okay, so these are some of the channels that you can get access to EMBL, and you can yeah, perhaps look it up. Okay, so now I wanted to show you a little bit about where I really work. So one of the units, which is EMBL in Grenoble, which is located beautifully between 3 different mountain ranges in the footsteps of French Alps. So we are located right here, a small institute in between the synchrotron of ESRF and nuclear reactor of ILL. And so I'm running my group there. We are about 5 people at the moment. Half of the group is working on the pre-mRNA splicing, so this kind of experiments which I showed you earlier. And the other part of the group is working on different aspects of RNA processing. So I showed you this different splicing cycle, and I just want to mention very briefly what kind of research we are doing at the moment and what really is exciting for us. So that was a splicing cycle, and what we are looking at the moment at is we are looking at the assembly intermediates also. We are looking at the biogenesis of some of these individual building blocks of the spliceosome. So we are focusing on the events which are happening much earlier before the whole spliceosome is formed. And to that end, we really want to understand the role of some of the factors which are assembly chaperones of this machinery, as well as we want to understand the order of events which are happening during this whole process. Okay, I'm sure Monica is going to show this slide, so I didn't want to steal it. But we have excellent programs for postdoctoral and also for PhD students, and you will hear from her in a second. And this really is to emphasize that so these postdoctoral and PhD programs at EMBL are not the same as EMBO fellowships. I'm sure Monica will also emphasize that. But to that end, so for example, my group has different partnerships with different groups at the EMBL, and we are actively recruiting people to do interdisciplinary projects which are based on cryo-EM and different kinds of single molecule microscopy and spectroscopy. And so if you are interested in this kind of stuff, please come and talk to me, and I will be happy to answer any questions. Okay, so with that, I would like to finish, and I'm happy to take any questions. Okay, thank you.
And now let's move to our next speaker, and our next speaker is Monika Lachner, and Monika is Head of International Scientific Training and Dean of the Graduate Studies at EMBL Heidelberg, and she will talk about international PhD program at Heidelberg and also about postdoctoral program and other opportunities at EMBL. Please, Monika.
Okay, all right. Okay, good, excellent. So where do I press for the next? Just here? Okay, I'll try. Yeah, well, I'm very happy to be here. And as already pointed out, so I will give you an overview about our training at EMBL, and it will particularly focus on the PhD program and the postdoc program. So I'm actually quite curious, within the room, how many people are here who are potentially interested in doing a PhD? Okay. And how many interested in doing a postdoc soon? A few more. Okay. All right. So, but I will cover both programs. All right. Yes, just to quickly give you an overview over training at EMBL, I will set the stage and then we go into more details. So this is how we are set up. At EMBL, we actually have a training unit. We are very similar to a scientific unit, meaning we have a head of unit, Anne Frossi, who is also one of our senior scientists. And we are organized in the EMBL International Center for Advanced Training, or short ICAT. So what we do at ICAT is we provide training for the next generation of scientists, and we do this via an internal training arm, And now I just have to see where is the here. Via an internal training arm, so this is actually my team, and via external training. So I'm not going to say much about external training today. This team is run by my colleague Jürgen Decker, but I think you can also find a lot of information online. The external training team organizes the EMBL course and conference program, which is different from the EMBO courses, but also slightly overlapping. So all EMBL courses and conferences are taking place in most of them are taking place at EMBO Heidelberg in the Advanced Training Center, and you can find a list of courses on our website. In addition to that, external training also runs the visitor program. I think this might also be interesting for you. So there is the possibility to visit EMBL, and you can do this at all career stages. We welcome internships, we welcome visiting pre-docs, we welcome visiting postdocs, and you can also, if you're a group leader, you can do a sabbatical with us. So I mean, do keep that in mind. Right. So in terms of the internal training, I have a team of currently 5 people, and we take care of 210 PhD students and 260 postdocs. This is our fellow population, and in addition to organizing the programs, we also organize complementary scientific skills training for them. Well, I will get started with the PhD program to give you an overview over that. In the EMBL International PhD program, as I said, we have approximately 210 students from 50 different countries. We have a long history. We are not quite as old as EMBL, but we were established approximately 9 years later, 1983. And we were one of the first structured PhD programs in continental Europe. So we have a long history in student training. We accept approximately 50 to 60 students per year into our program, and we run 2 international recruitment rounds. I will also show you a little bit more about them and when they take place. Since 1997, we are actually allowed to confer our own PhD degree. However, up to this point, we have mainly conferred degrees as joint PhD degrees in collaboration with partner universities in the member states. Now, this is our student body in 2018, end of 2018. So if you are good with geography, you can see we do have Polish students. At that point, we covered 48 nationalities. 46% of our students were female. But this is actually really, I mean, when you look over the years, we are approximately 50%. I think this is a kind of a fluke that we are lower right now. The site distribution I also wanted to mention. I mean, we really take care of all of students at all the different sites. So the majority of our students are located in Heidelberg here, but you can see we also have students at the other sites. Next biggest one would be the EBI. In Barcelona, we only have 1% of our students, but this is really due to the fact that only the first students started in 2018, so this number is definitely going to go up. Now, how do we recruit? And I think now that will be interesting for anyone who wants to apply for the PhD program. We recruit twice a year, as I said, in order to be eligible. You need to have a degree that would allow you to pursue PhD studies in the country where the degree was obtained. So if you have a UK degree, you could have a bachelor. If you're from Germany, you need a master. You need 2 reference letters. This is actually really important. So make sure if you apply, really make sure that your referees submit the reference letters. We have an online application platform. It's actually quite easy. So you log in, we ask you all the typical questions about your academic background, and we also ask you to write a few essays on different aspects. Now the evaluation, I think this is also important for you to know. So we screen all applications, we evaluate all eligible applications, and this is done in a 2-stage process. In the first stage process, all faculty is involved. So, right, Wojtek, you sometimes have a lot of applications to look at. And then in the second stage, we actually do the evaluation by the individual research units, and based on that, we then have a short list for candidates to be interviewed. At the interviews, what we do, what you could expect is a panel interview. So this is an interview that you need to pass to be eligible for the PhD program, and then there are follow-up interviews with group leaders, and then at the end, of course, we need to do the matchmaking, which is, of course, the most important step in the entire process. Now, if you're considering to apply, these are the upcoming recruitments. So the Spring Recruitment 2020 will open the application platform in August. And we also have a summer recruitment in 2020, and there the application platform will open in February 2020. In terms of student funding, I just want to highlight one point. Our PhD positions actually do include Social Security, I think because this was an important point that was raised. So you will have health insurance. So we have a really good package, so no worries there. It's a really, really good deal. What do we think is important for PhD education? So we have several topics that we consider as key features of our program. Maybe to start here, I think this is fairly state-of-the-art for PhD programs these days. We have an extensive mentoring system that includes a thesis advisory committee. I think you are all familiar with this. And I think the great thing about most of our groups is that we have a very good pre-doc to supervisor ratio, so you can get really good supervision and like close supervision if you want that. In terms of the training curriculum, we offer a broad interdisciplinary training. I'll show a little bit much more how we do this in the next slide. Transferable skills training. We now have a career advisor for our PhD students because we really think that it's important to make informed decisions about your career. We strongly support early independence, and this we do, for example, by giving our students the opportunity to organize an annual PhD symposium, and that's entirely organized by them, so we don't interfere with this. This is the next one on the list, so if you want to apply, do. It's a really great opportunity to meet other students and to network. In addition to that, of course, we also have PhD retreats and a very active pre-doc representative community. The partner universities I already mentioned, so we really try to allow people to keep their links to their home countries. I mean, this was also one of the points that came up in the earlier discussion. Now, the EIPP student life cycle. So what I wanted to show you is actually one of the things that's very specific about our program is we have a starting pre-doc course. And what it means is that all the pre-docs from one class get together and they are spending 9 weeks in Heidelberg. It's a very intense course, and when I talk to alumni, they often tell me, oh, the one thing that I remember most of my PhD is actually going to the pre-doc course. I think it's a great instrument of building a cohort and also building networks. So the pre-docs at EMBL are actually the groups that are best connected within the entire institute. And then basically what we expect you to do is to try to finish your PhD within, or submit your thesis within 4 years. And during that time, you have to do your annual tax and work on your career development and your scientific training, and of course also participate in courses and conferences. Now, how does it work out? So this is the actual numbers. So the time to submission is currently 3.9 years. So most people actually manage within 4 years. Most of our students who start with us do submit. So we have a submission rate of 95%. I think the publication record, I think, is something that many people ask about. So currently we have 1.3 first author papers on average, meaning some people have more than one, but we also occasionally have a student who doesn't have a publication when they leave. I mean, that's, you can see in here. But overall, our pre-docs are very successful, and we recently did a big study in terms of like tracking them, where they are. So we do follow up with them, and we also frequently invite them to our events. So we have a good idea of what they're doing. So 63% continue in academic research, so really the majority does a postdoc, that's very clear. We have approximately 15% in industry research, 16% in science-related positions, and then 6% in non-scientific careers. And I will show you later on how that compares to our postdoc population. Right, so I thought maybe we do questions now, if there are questions that are specific for the PhD program.
What's the success rate for the applicants?
Good question. So we have, as I said, we have around 60 positions each year, and we have 1,800 applicants. But but let me put that into perspective. So if you're invited, you have a 30% chance of getting an offer on average.
How many Polish students are?
Currently we have 8 Polish students in the PhD program, and we always have a good representation of Polish students. I mean, I have to say, but it would be good if we would get some more applications from Poland. So that's also one of the reasons I'm here. Oh, and I want to say, you know, the thing with applying, and I think that, I mean, it's true for the long-term fellowships as well as for the PhD program. If you don't try, you cannot win. Okay, so to the postdoc program. There things are slightly different. Now for our postdoc program. I already mentioned we have a steady state of around 250 postdoctoral fellows at EMBL. They can stay a maximum of 5 years. So usually what you get is you initially get a 2-year contract. So this is the minimum time of postdoc contracts that we give out. And then this can be extended up to 5 years if the funding is available. The postdoc community is a much more heterogeneous community than the PhD community. That's clear because they come in via different entry routes and of course also via different funding schemes. And I think this is really important for you to keep in mind. There are several ways of getting to EMBL if you want to do a postdoc there. And I'm mentioning these 3. So, I mean, the first one is the classical postdoc stream. So this is what you would do if you wanted to go to any other institute. You contact the group you're interested in, you point out to the group leader why you really would like to work with him or her, and then one looks into the funding. So most of our postdoc positions are funded via third-party funds from the group leader or via person in merit fellowships such as the EMBO long-term fellowship, HFSP, and so forth and so forth. This is approximately 30 to 35 people who come in like that every year. Then we have a specific program that's a centralized program where we actually also have 1 call per year, so you can apply for this program. And this is the EMBL Interdisciplinary Postdocs program. There we have an intake of 20 people per year. And again, this is a program that has deadlines where you have to apply at a certain point, and then we have interviews at a certain point. I'm showing more about that in a minute. We also have, and this is actually growing, so I would encourage you to check that out as well. We also have site-specific programs that are modeled on the iPod program. These are smaller programs. They have approximate intake of 2 or 3 fellows per year. For example, the EBI has a collaboration with Sanger and a collaboration with the BRC in Cambridge. And just recently, and this is the first year that this is open, this is a site-specific program in Italy between EMBL Rome, the IIT Genoa, and the EBI is also involved there. So, I mean, this is also worthwhile checking that out. Right, so now let's talk about the iPod program. I think the iPod program is really our flagship program. It was established in 2007, and there the point was that on the one hand, we wanted to foster collaborations within EMBL, and on the other hand, we wanted to provide high-quality interdisciplinary research training for fellows. And what we ended up with was the iPod program. So the iPod program works in a way that a fellow actually has to work with a minimum of 2 EMBL groups on an interdisciplinary research project. This is the core of the iPod program and will always be the core of the iPod program. Since 2007, the program has evolved quite significantly. I'm not going to go through all of this in detail, but it has evolved also that much due to the funding situation. So we were very successful with the co-funding actions under Marie Sklodowska Career Actions. So basically, we had 2 successful applications under Framework Program 7 and now 2 under Horizon 2020. And what the Commission encourages you to do is to put forward innovations and really introduce new benefits for the fellows. And that's why the program has changed a little bit over the years, but the 2 important points, internationality and collaboration, remain the same. So, and I mean, this is just to show you that it is a very international program. Again, here we also have, Poland is covered under the iPod Fellows. This is to show you, this is 60 fellows representing over 30 nationalities. And in terms of collaborations, this is a collaboration network to show you that the different units really are working with each other on these projects. So it has been a very successful program for EMBL, but also for the fellows. Now, this is the most recent incarnation of the iPod program. It's called iPod 4. And Wojtek has already shown you the advertisement slide. So this is the most recently funded program. So we just started this year with it. And in iPod 4, the framework is as such. Every iPod 4 research project has to fulfill 3 criteria. It has to be interdisciplinary, a minimum of 2 EMBL group leaders has to be involved, and it has to be a self-designed project, meaning the project idea should come from the fellow. So there are no pre-designed projects. The way it works is, if you're interested, you get in touch with Wojtek, for example, and you discuss your ideas with the group leaders, and together you can then shape the project. What is also possible with an iPod 4 is that you can have an external research partner. This is a collaborator who's not at EMBL, so who's outside of EMBL, and there are funds available to spend 6 months at the external research partner lab. And depending on which sector you're going to, you can either have an academic research partner, then you're in the academic track, or an industry research partner, or a clinical research partner. So the clinical track is actually really new. And what we are trying to do with this is to, this should attract fellows who are interested in biomedical research, in translational research, and we also hope that we will be able to attract some MD PhDs who would like to become physician scientists. So this is the idea of the clinical track. And we'll see how it works, because this is actually really new, so we have never done that before. It will be interesting. In terms of the benefits for the fellow, you will receive a 3-year contract. So this is different to the regular 2-year contract at EMBO. So 3 years is pretty good already, I'd say, for a project. In addition to that, we provide a training program that I'm now not going to go into detail. It's a 6-day training program. We have a pretty big public engagement activity, where we will work with the Natural History Museum. So we are super excited about that. So they will do a public engagement workshop with the fellows, and then the fellows will actually do an activity in the museum. So this will be really, really cool. We put big emphasis on career development. We have a career development plan, a career secondment scheme where you can actually check out careers that you may be interested in. and we will also offer some mentoring opportunities. Plus, this is also the first time we are offering conference funds. So it's a pretty good package, and I would really strongly encourage you to check out the webpage and consider to apply. Well, that's now the application. This is the official call slide. Actually, the call opens on Monday. So on Monday, you'll find everything on our web pages, and you can apply until September 5th. I just wanted to mention, and this is I'm almost at the end, in addition to the activities that we do for the iPod Fellows, we of course also provide a lot of training for the remaining postdoc population. We have a second mentor program. So every postdoc at EMBL is encouraged to get a second mentor from EMBL faculty. So the idea is that there are informal meetings. In these informal meetings, they can discuss scientific questions, but also questions that relate to career development. We have a career service, I already mentioned that. A pretty big training pool of training opportunities, be it grant writing, scientific presentation, lab management, so anything you need as a successful scientist. And we also do interview training, so this is actually quite popular. So every year we do 2 preparing for the academic job market workshops, and this is essentially mock interviews for late stage postdocs who are about to apply for group leader positions. There are pretty tough questions that are asked in these workshops, but I think it's always better to be asked tough questions by your friends than when you actually go to the interview and you don't know what to say. So it's actually quite, our fellows, I think, really benefit from it. And they also mentioned that for some of them it was an eye-opener because there were questions asked that they didn't expect would be asked in interviews for group leader positions. We offer, in the past years, we have also offered a similar workshop for industry positions, which is also quite popular. Now, in terms of career destinations for EMBL postdocs, this is again a result of the career study that we've recently done. And basically, when we look at our alumni population, you can see that around 30% of our postdocs are principal investigators now, which is a pretty good rate. But you can also see what we have seen over the years now, that there's an increase in other academic research and teaching roles, so especially in the heads of facilities. So we see an increase in these positions, probably also because they are now available at many institutes. Right, and with this, I'm at the end, and I just wanted to actually show you the people who do a lot of the work. So it's not me doing the bulk of the work, actually, it's my team. So this is the graduate office. This is the iPod team. And if you are considering to apply for iPod, so Brenda will be the person who would be answering all of your emails. So now you already have a face to the name. And these are the email addresses that you should use if you have any specific questions. And you can ask me in the coffee break, of course. Or now.
I think we have time for questions, so. About the opportunities.
Well, I'll be around, so if you want to talk. Yeah, okay.
A few questions.
Similar to the previous question, what is the success rate for postdocs?
Well, that's actually, the thing is what we see there, and that's quite interesting, is that we have a lot of people who open an application, but we don't have that many who actually submit. And I think the reason being is that it's actually a quite, it's a process that requires quite some proactivity. You need to get in touch with your host, and I think sometimes people just give up at that point. So, no, really. I mean, I actually. So we invite. I can tell you how it is. We invite around two times as many candidates as we have positions. So if you're invited, you have a really, really good chance of getting the position. And I think really do apply, because as I said, there are not that many people who submit their applications in the end. So that's, that's important to know. Be persistent, and if group leaders don't react to your first email, just follow up with a second one, because they may have overlooked it or they didn't have time to respond at that time.
So, Monica, I wanted to ask you, because EMBL can award a degree, PhD, and you organize, you create joint programs with universities.
It's not joint programs, it's joint degrees.
Joint degrees, yes.
So
my question would be, what conditions the university needs to fulfill?
Well, okay, so basically what we do is we usually, I mean, the rough guidelines are that we say, okay, usually one university per member state. We usually say it should be a university where we have a lot of students, where a lot of our students are from, or where we get a lot of applications from. And then, I mean, we have certain, let's say, requirements that are important for us. And these are requirements that we then negotiate with the university, and we end up with an agreement where everything is regulated in terms of, like, you know, what is needed for registration, what is needed for submission, and so forth and so forth.
Is there any Polish university involved in such?
At the moment, not.
No. Do you have further questions? If not, well, now it's coffee break. So thank you for your talk.